Good morning to all of you. Good morning, children. So yesterday we started with the next chapter, which is respiration in plants. So in that I told you what happens in anaerobic respiration. Yes, I'm getting your good morning messages. Good morning to all of you again. So let's start with a topic, respiration. I told you what happens in respiration. I told you that the anaerobic respiration, I told you about anaerobic respiration, that in anaerobic respiration, the glucose undergoes the process of glycolysis, and by the process of glycolysis, what is formed is pyruvic acid, and then this pyruvic acid which is formed, these are two molecules of pyruvic acid are formed because pyruvic acid is a 3-carbon compound. It is a 3-carbon compound, and glucose is a 6-carbon compound. So what happens that only if 2 molecules of pyruvic acid will be produced, then only those 6 molecules, 6 carbons will be utilized, because 3 2s are 6, 3 2s are 6. So when two molecules of pyruvic acid are formed, then only the 6 carbons which are there in CO2 will be adjusted here. See, now this is 6 carbon. So 2 of the pyruvic acids are formed, and then this pyruvic acid, which is formed as a result of glycolysis, it has 3 ways to get used up. And what are those 3 ways by which the pyruvic acid gets used up? The first way is the anaerobic respiration. In anaerobic respiration, that is in the yeast. What happens in the yeast and what happens in some bacteria and muscle cells. So in yeast, this forms, the pyruvic acid forms ethyl alcohol or ethanol, C2H5OH is formed. Now, how many molecules of C2H5OH will be formed? Because C2H5OH is a 2-carbon compound. As you know, it is a 2-carbon compound. So two molecules of ethanol will be formed. Now, till now, when 2 molecules of ethanol are formed, 4 carbons are used up. 2 2s are 4. So 4 carbons are used up, and still 2 are left. Still 2 carbons are left, because how many carbons in pyruvic acid? 6 carbons, because pyruvic acid is a 3-carbon compound. One pyruvic acid has 3 carbons. And these 2 carbons, when ethanol has 2 carbons and 2 molecules of ethanol are produced, so that way we see that 4 carbons are used up, and still 2 are left. Still 2 carbons are left. So those form carbon dioxide and 2 molecules. So 2 carbons here and 4 carbons here. So it makes 6. And here it is a 3-carbon compound. So 2 of it is formed. So 2 into 3, 6. Here also 6, and here also all 6 carbons have been adjusted. So this way, this happens in yeast. And what happens in case of in some bacteria and muscle cells? This pyruvic acid, it forms lactic acid. The formula for which is C3H6O3. This is the formula for lactic acid. I told you this formula yesterday also. But if you see, one molecule of lactic acid has 3 carbons. So how many lactic acids will be formed? This has 6 carbons. Two molecules of pyruvic acid has 6 carbons, and here we see if only one lactic acid will be formed, it will adjust only 3 carbons. So 2 lactic acid molecules are formed here. Now 6. So 6 carbons have been utilized. If molecules are formed, and that way what happens that we get all the 6 carbons which are here, they get adjusted here. Now this was about anaerobic respiration. Now I'll tell you about aerobic respiration. So in aerobic respiration, what do we see? So aerobic respiration, how the glucose molecule is used up. As I told you before also, glycolysis is the common pathway for whether it is aerobic or anaerobic. Glycolysis is the common pathway in both the type of respiration. So here what happens, the glucose molecule undergoes the process of glycolysis, and this glycolysis, the product formed enters another cycle, which is Krebs cycle. So after glycolysis, it enters the Krebs cycle, which takes place in mitochondria. Then the product formed enters ETC, which is electron transport chain. And this electron transport chain takes place in the inner membrane of mitochondria. And after this, the product formed enters the oxidative phosphorylation. And this oxidative phosphorylation, which is the last step of aerobic respiration, this takes place in F0, F1 particle. You remember what is this F0, F1? We have done it in one of the processes. Do you remember what is this F0, F1? Anybody who wants to answer this question? F0 and F1? Yes, beta? Yes, bache? If you remember this? Anybody? Yes, beta? ATP synthetase. Yes, Amrita is saying ATP synthetase. That's true. Yes, these are the two parts of ATP synthetase, and we studied this in chemiosmotic hypothesis. So this is aerobic respiration which takes place. Now we come to the first process of respiration, which is glycolysis. So we will start with glycolysis. As I told you, glycolysis is the common pathway. It is the common pathway for aerobes and anaerobes. Common pathway in aerobic and anaerobic respiration. This is a common pathway in both anaerobic and aerobic respiration. The second point, it takes place. Where does it take place? Takes place in cytoplasm. So this is the site where it takes place, that is cytoplasm. This process of glycolysis is also known as EMP pathway. This is also known as EMP pathway. And why this is known as EMP pathway? This is known as EMP pathway because of three researchers who discovered it in a way. And the name is Amden, Meyerhoff, and Parnas. Meyerhoff, and the third name is Parnas. Because of the names of the discoverer, this has been named as EMP pathway, or Amden-Meyerhoff Parnas pathway. Now, how does this pathway goes on? What happens during this pathway? See, if it is an animal cell, the glucose directly enters the process of glycolysis. If it is an animal cell, glucose directly enters the process of glycolysis. But if it is a plant cell, then it will have sucrose in it. So in case of plant cells, the sucrose first gets converted into fructose and glucose. And then this glucose enters the glycolysis. This enters the glycolysis. This happens in plant cells. So this is how the process of then glycolysis takes place. It is a almost 10 steps pathway. And ultimately results in the formation of some ATPs. Almost 8 ATPs are formed. Results in the formation of 8 ATPs are synthesized. We can write here. Eight ATPs are synthesized during the process. So this is the way it goes on. Now I'll tell you how this process takes place. For this, I'll erase it. So now what happens during the process of glycolysis? First of all, the glucose which enters the process of glycolysis, this utilizes one ATP. When ATP is utilized, it breaks down into ADP. So one ATP molecule has been utilized, and by utilizing this one ATP, what happens that an enzyme is also used. So here one enzyme you can write down, hexokinase. This hexokinase is also known as phosphorylase. So you can write down anything. You can write down hexokinase, or you can also write down phosphorylase. And this results in the formation of glucose 6-phosphate. So here by this glucose 6-phosphate, I'm just writing PHO. Okay, here once I'll write full and then I'll write only P. So glucose 6-phosphate is formed. Now after the formation of glucose 6-phosphate, see, this is formed by utilizing one ATP. Here if you see, one ATP has been utilized. Now this under the action of another enzyme, and the name of the enzyme. So another enzyme isomerase is used, and in the presence of this enzyme, the glucose 6-phosphate gets converted into its isomer. Now what is the isomer of this? So glucose 6-phosphate gets converted into its isomer, which is fructose 6-phosphate. In the presence of enzyme isomerase, the glucose 6-phosphate gets converted into its isomer, which is fructose 6-phosphate. Now, this fructose 6-phosphate utilizes another molecule of ATP. One more ATP is used up here. So ATP gets converted into ADP here. Because one molecule of ATP is used, it gets converted into ADP. And again, wherever ATP is used, we will write the enzyme hexokinase or phosphorylase. So I'm just writing HE or phospho, the first enzyme which I have written. Hexokinase or phosphorylase, the same enzyme we'll write here. So by using this, it gets converted into fructose 1,6-biphosphate. So this is fructose 1,6-biphosphate. Fructose 6-phosphate gets converted into fructose 1,6-biphosphate. Now this fructose 1,6-biphosphate, it undergoes another breakdown in the presence of one enzyme, and the name of the enzyme is aldolase. Splitting takes place here in the presence of the enzyme aldolase, and after splitting, it forms two compounds. One is dihydroxyacetone and another is glyceraldehyde phosphate. So dihydroxyacetone and another thing which is formed is, here I'll write glyceraldehyde phosphate. This dihydroxyacetone can get converted into glyceraldehyde phosphate, or itself this glyceraldehyde phosphate, it undergoes further process, or this may get converted into this and the further process takes place, or directly glyceraldehyde phosphate will continue with the rest of the process of glycolysis. Now this utilizes, this makes, just a minute. Yes, now this glyceraldehyde phosphate, it undergoes a change. Here I'll write it here first. This name I'm writing up here for your convenience. Takes up inorganic phosphate, takes up inorganic phosphate and synthesizes from NAD, is formed NADH twice. NADH twice is formed and it results in the synthesis of 1,3-biphosphoglycerate. And what is formed? 1,3-biphosphoglycerate. This is formed 1,3-biphosphoglycerate. Now, the next step is formation of 3-phosphoglycerate with the release of ATP. In the next step, ATP is formed. From ADP, ATP is synthesized. So see here 2 molecules of NADP are synthesized, here 2 of ATP are synthesized, and from 1,3-biphosphoglycerate we get 3-phosphoglycerate. glycerate or glyceric acid. 3-phosphoglycerate. Then after this 3-phosphoglycerate under the action of another enzyme, which is mutase, or you can say phosphoglyceromutase. It synthesizes 2-phosphoglycerate. Phosphoglyceromutase. Phospho is this, glyceromutase. It forms 2-phospho. So from 1,3-biphosphoglycerate, 3-phosphoglycerate, from 3-phosphoglycerate, 2-phosphoglycerate is formed. Now the next step is again under the action of another enzyme, which is enolase. Under the action of another enzyme, which is enolase, ENOLASE. What is formed is phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate is formed. And the next step is, the last step of this is synthesis of ATP. From ADP, ATP is synthesized. Again, 2 molecules of ATP are synthesized, and enzyme will be the same, hexokinase or phosphorylase, and it results in the formation of pyruvic acid. It results in the formation of pyruvic acid. So finally, in the process of glycolysis, this is the process of glycolysis. This is the process of glycolysis. So in the process of glycolysis, finally, what do we get? The end product is pyruvic acid. So if you see, these are 10 steps. See, the glucose utilizes 1 molecule of ATP under the action of enzyme hexokinase or phosphorylase and results in the formation of glucose 6-phosphate. This under the action of isomerase gets converted into its isomer, which is fructose 6-phosphate. Again, this fructose 6-phosphate utilizes another molecule of ATP, which gets converted into ADP, and under the action of same enzymes, hexokinase or phosphorylase gets converted into fructose 1,6-biphosphate, which undergoes splitting under the action of aldolase. So this gets split under the action of enzyme aldolase and gets broken down into dihydroxyacetone and glyceraldehyde phosphate. Either this dihydroxyacetone gets converted into glyceraldehyde phosphate, or itself glyceraldehyde phosphate then further carries out the remaining steps of the glycolysis. So this then carries out further remaining steps of glycolysis, in which this glyceraldehyde phosphate makes up one inorganic phosphate and makes NADH2. This is high energy compound. Another high energy